So in this video, I'm going to discuss what is HIV and why is it so scary that I would put myself through a month-long treatment of drugs that made me basically stupid. I've actually had a nut job channel called HIV Answers troll my page linking HIV testing with Dick Cheney, saying I shouldn't have had the drugs. I wanted to talk about the side effects first, but I need to get this part out of the way first, sadly. In 1980, young men from the gay district showed up with pneumocystis harnii, pneumonia. It's a respiratory disease that shows up in compromised immune systems only. Soon, many had Kaposi sarcoma, which is a very, very rare skin cancer only present in old people. It became known as the gay cancer, or AIDS, acquired immune deficiency syndrome. In 1983, two different teams discovered a retrovirus that was present in all AIDS patients, and HIV was the name chosen as they both had competing names for the virus. After that, big efforts were made to raise awareness of the disease, and it turned the gay community on its head. The gay community in that time period was extremely promiscuous, but at the same time, so was the straight community. Today, much of the gay community has turned into some of the biggest champions of safe sex education. During the Reagan years, since AIDS was just a gay problem, getting funding for it was like pulling teeth, sadly. However, we slowly got over that stigma, especially when Brian White got it from a transfusion and people began realizing that it was a straight problem too. In the 90s, we made some great progress in treatment thanks to this newly raised awareness. And from that research, our understanding of genetics actually exploded. Thanks to that, at this point, HIV is now a long-term treatable disease so long as you have money to pay for it, which many Americans and most Africans don't have. So what is HIV and why is it so hard to treat? Well, the central dogma of biology states that DNA gets transcribed to RNA by a protein called polymerase. It's where you see the A line up with a T and the G line up with a C to make a copy, although T is replaced with U, when it becomes RNA. But that's an extremely deeper topic. You've seen it in high school. Assuming you're in the Y generation at least. After that, RNA leaves the nuclear membrane and is translated into a protein strand by an enzyme called a ribosome, which, using a lock and key-like mechanism, reads 3 base pairs called a codon and adds an amino acid to the chain for each 3 base pairs. Certain combinations of base pairs tell the ribosome when to start reading, and some tell when to stop. When the protein chain is released, it then folds itself up, many times into more enzymes, but the structure is determined by the charge and bond forces on the chain. Temperature and pH may also change the structure as well. A mutation in DNA or error in the RNA can have either a negligible change on the protein structure or it can turn into something completely new, like the enzyme nyelinase that was a mutation of some other enzyme that through mutation in a fungus can now gain nutrients off of nylon. For more information on the folding properties of proteins, you can go to the game Fold It that actually helps scientists figure out how a certain protein chain will fold most efficiently. So what does this have to do with HIV? Well, the central dogma of DNA to RNA to protein was long held. No one ever thought that the process could go backwards. Turns out HIV has something called reverse transcriptase. It goes into the body as a strand of RNA and converts itself into DNA. Then it uses an enzyme called a transposase that commonly moves pieces of DNA called transposons in your cells at random. This can lead to mutation, so it puts itself into your own DNA to hide from your immune system not doing anything. A normal virus, be it DNA or RNA, comes into your body and hijacks your polymerase, if it's DNA, and your ribosomes, and uses them to copy itself until the cell runs out of resources. Then it explodes via apoptosis and infects other cells. Usually, after enough time, the immune system can catch the infected cells and tell it to kill itself via apoptosis. That's what apoptosis means. It's cellular self-destruction. As HIV is hanging around as part of your DNA, it doesn't get detected. It's not until that cell becomes damaged that it begins attacking the cells and replicating. On top of that, it also has a nasty habit of entering your immune system and killing off your T helper cells and macrophages, which are essential to the functions to killing off invaders. Immune cells are actually the primary infected cells, but there is also evidence that HIV hangs out in the gut cells and lays low, so the immune system is constantly playing cat and mouse. What my drugs did for me was to deactivate reverse transcriptase so that the RNA virus could not convert itself into DNA and stick itself into my chromosomes. It may also deactivate other enzymes, which is probably why I was so draggy and my immune system so hyperactive when I took it. This is just one of the few drugs used by patients with HIV that they have to take for the rest of their lives, but thanks to the drugs, it can actually be a long life with just side effects. The fact that it hides out so well is why it's so hard and at the moment near impossible to cure. There's some evidence that certain bone marrow transplants might actually be able to cure it, but it's a very harsh treatment. I had someone ask if, since I was taking the drugs, would it mask whether I had HIV or not in tests. No, HIV tests are actually not testing for the virus. What they are testing for is the specific antibodies your body is making in response to the presence of the virus. At first, your body can kill off infected cells really well, but the cat and mouse game over time weakens the immune system to the point that it can't work anymore. I'm at the 1.5 months mark, and nothing has shown up. And if nothing has shown up at the 6-month mark where the HIV antibody would be at its highest, I should be given a clean bill of health. It's a bit unnerving, but science and the odds are very much on my side. So lastly, we ask, where did this disease come from? Well, as far as we can tell, this disease was first transmitted from uncertain primates to humans in the 1920s. SIV, or simian immunodeficiency virus, exists throughout the primate family. A primate species is usually completely immune to a native strain through millennia of weeding out the ones without resistance mechanisms. However, when one SIV strain gets transmitted to another primate species, it becomes AIDS. And no, we didn't get HIV from sex with monkeys, even though that is the more scandalous version. We got it most likely from eating them. Someone probably cut themselves during the butchering process, and blood to blood is the easiest form of transmission. It's why we took my accident so seriously. HIV is highest in gay men here in the US, but it's highest in heterosexuals in third world nations. The main reason is because they have many other diseases like other STDs or infections that increase likelihood of transmission. Also, rape being used as a weapon of terror is spreading it even more. The Pope, of course, just a few years ago told the people of Africa that condoms cause AIDS, even though studies show that proteins in semen increase infection rate by 200,000 times, pretty much attempting to condemn half a continent to death with misinformation. Just like with any other area of science, there are denialists, and there are huge AIDS denialists in position of power, especially in African governments that want to have to spend less money on helping people with HIV. So how do you avoid HIV? Never share a needle no matter what. I know most of my viewers won't have that problem with this, but just in case, never let something that isn't completely sterilized penetrate your skin, especially around blood products. Of course, we all get cuts, but unless there is other blood, just some peroxide and Neosporin will work for that. Also, use condoms if you aren't with someone you haven't been monogamous with more than 6 months and have not been tested. 6 months is the time that it takes for antibodies to build up, so save bareback for people you are with for quite a while. While the infection rate for oral sex is a fraction of that of penetration on men, and even half of that on women, it's recommended that oral sex use barriers as well. Don't try and clean up body fluids without proper training. Bleach kills almost anything, and is often used in the cleanup of body fluids. I hope this helps some people better understand HIV and the issues involved with it.